So this review is from Nature Reviews, Microbiology, published in October 2020. And the title is Considerations for Diagnostic COVID-19 Tests. And the authors are Olivier Vandenberg, Delphine Martini, Olivier Rochas, Alex van Belkom, and Zizis Kozlakidis. And they are from 3 different countries, primarily from Belgium. And then there is one person from the University College London and 2 individuals from France, including one from a WHO lab. And the reference, this is quite a robust article with 176 references, of which 168 or so are research articles. And they have cited 3 WHO websites, or web pages rather, 2 WHO technical guides, 2 UNIDO articles, and 1 magazine article, but that also from the Harvard Business Review. And I did get a few complaints saying this is very difficult reading and it's very complicated. So I will try to make it as simple as possible. So in this review, to present you the abstract, they simply summarized the crucial role of diagnostic tests with during the first global wave of COVID-19. And as we all know, we are in the second wave now. And just to, I thought I will just present you the latest numbers. As of 9th February, this is from the WHO website, we have 105.4 million cases reported with 2.3 million deaths. So that's how devastating this pandemic is. So the crucial role of diagnostic tests, and then they explored the technical problems encountered within developing and the rest of it of diagnostic tests, and of course implementation. And then they go on to define future directions for the better use of diagnostics, where they actually predict possible resurgence. They're talking about second waves, third waves, fourth waves, and so on. So I hope we will not go into that. And thirdly, they suggest that there should be continuous global improvement in diagnostic tests for more rapid detection of patients, possibly at the point of care, wherever they are, the hospitals or wherever the isolation centers or wherever. And that would lead to optimized prevention and treatment. What I like about this review is they talk about both industrialized countries and low resource settings, such as in a developing country such as ours. So that is the simple abstract. Now in the introduction, they talk about multiple parallel clinical priorities during a pandemic. So they say that there are 3 basic areas that scientists should understand first. Firstly, the symptoms and the pathology of the disease should be understood. So again, symptoms are manifested or shown when the pathology is active. Pathology is when the virus, in this case, interacts with, say, our immune system and invading our tissues and interfering with our physiology and causing damage. That is the underlying pathology. So when there is pathology, that is manifested in the form of symptoms. So we will have a cough, a sore throat, or perhaps the voice, there's a change in voice, change in taste, smell, fever, and so on. So those are the symptoms. So understanding symptoms and pathology would lead to optimized patient care and also prevention of future. We will know how to prevent future infections. For example, when we knew that this is the mode of transmission of the virus via droplets, via nasal secretions, and so on, then came the masks and the distancing and so on. And secondly, these are parallel clinical priorities. They talk about diagnosis to characterize and detect or diagnose the causative agent, in this case the virus, or say immunological consequences. For example, when the virus gets into our body, our immune system will recognize the virus as a foreigner that is carrying foreign proteins, and our immune system will mount immune reactions towards this foreigner, this visitor. And they can be, the simplest form will be to mount antibodies that everybody will understand. So what they say is we should be able to diagnose the causative agent and to look at such things as antibodies and antigens uh, as a starting point. Then moving on, we should be able to define the pathogen both biologically and genetically, and whether to see whether it is inducing any immunity in us, maybe protective, maybe not protective. Protective is our immunity itself will protect us from the pathogen or the virus in this case. So we have to know about all those things because these are the points that are key to the development of vaccines and therapeutics. So these are the three parallel uh, clinical priorities that one should uh, have or put down during a pandemic. Now I'm going to go off this serious, this complicated mode, and I thought, although they have not, they have referred to many papers which give out uh, all possible information about the virus and how the diagnostics were developed and the good and bad of those and so on. I thought I will give you a very simple introduction to the virus and then how knowing the virus and its genome and its proteins led to the development of the diagnostics that we all are aware of. Now, this is a picture, and I don't think it's a true one, but the diagrammatic schematic picture, which shows that these have spikes coming out from the surface of the virus. And because this is not only for SARS-CoV-2, but all coronaviruses have this characteristic, and they were called coronaviruses because of these special structures that are jutting out, which was like crown-like. So another word for crown-like is corona. So therefore, this is the SARS-CoV-2 virus that we are talking about. So when we look at the coronavirus structure, very simply, we all know that it's an RNA virus, a single-stranded RNA virus. So we have the single strand of RNA here. And then on top of that, we also had to look at the proteins of this virus. So the important ones, or the very few proteins that are there, and of course the important ones are the spike protein, okay, that is there, the spike here, that is spiking out here, which is very important biologically because it is this protein that reacts with the receptors on our cells, on human cells, and that connection, that ligand receptor connection will enable the virus to enter into our cells. Therefore, the spike protein is very important. It's for short. Then we have the nucleocapsid protein denoted by N, the membrane protein by M, envelope protein denoted by E, and so this is the simple structure of the coronavirus. Moving on, I'm just going to show you these complicated pictures just to prove a point. I'm not going to go through these because these are very complicated, but scientifically all these things have been proven, and I'm going to take the liberty of showing you some very simple sketches which will be very informative, although not scientific, not exactly scientific. So please pardon me for that, but I want to make this as simple as possible. So these sketches are from New York Times, from a very interesting article that was there to make public awareness, to create public awareness. So now here, if the individual was first exposed to the virus, this is the initial exposure. and through so many days, after so many days, symptoms will appear. So the period between exposure to the appearance of symptoms is known as the incubation period, where we will not be aware that we are actually infected. So this was initially put down as 4 to 5 days, but when I checked up yesterday, it has shifted to 5 to 6 days. And it can last up to even 14 days. And in very rare cases, the incubation period has even gone up to 21 odd days. So that is in rare cases. So in a particular article, they say that 95% of the individuals in that population that they were testing had approximately median incubation period of 11 days, but this was in another country. So globally, this is the accepted incubation period. And then symptoms will arise. I'm just taking you through this axis. Symptoms will arise, and usually symptoms will last for about 10 days. All these are approximations because it can be plus minus depending on the viral load and your immune system and your general health and so on, age and so on. And then thereafter there can be recovery. So very mild infections, recovery is predicted in about 2 weeks, 14 days, whereas very acute or bad infections can have a recovery period of up to even 6 weeks, even later. So now the reason for our quarantine recommended for 14 days, if we think we have been exposed to somebody, is because to cover this period. Because initially it was 4 days. So approximately 14 days quarantine is recommended if we think we have been exposed to somebody positive. Then let's talk about the viral load. So there have been mathematical models that have been done, and these have indicated and recorded that the viral load, as you can see, will start going up with days of exposure and will tend to peak before symptoms appear. So it's only after peaking that symptoms will appear, that is the highest viral load, and then the symptoms will appear, and then there's a rapid decline in the viral load following the first signs of illness. And a person is supposed to be infectious during this high viral load carrying period. So all these are very important information for developing tests and using tests during the course of this viral infection. So coming back to the review, the current COVID-19 diagnostics, they say, are twofold. And these are the direct detection of the virus using molecular methods, that is to detect the viral RNA. And we are all aware of the RT-PCR that is being used. And secondly, under this category, detection of the viral proteins via the rapid antigen test. Then the other direction is immunological testing. And these are based on the detection of virus-specific antibodies. And we know that it is called the rapid antibody test. Now, so actually we have 3 tests that are available. And of course, there are different types of tests that are being developed to detect the viral RNA. I'm not going to harp on those. But I also sent a second paper, Use et al. 2021, which is also included in this review, because it gives a for those who want to read more about these different types of tests, they give a very explicit description of the development of these tests, the advantages, the disadvantages, and then they also talk about the new types of tests that are currently being developed. So now, with this background, when we talk about testing, it is said that the rapid tests, and here I have to emphasize that here it is the antigen the rapid antigen test that will detect a high virus load, whereas the PCR test will have a larger range of detection power. So it can capture even low levels of viruses, but not necessarily indicating that these people obviously will not be infectious, right? So the rapid tests you will see will capture the infectiousness or infectious individuals. Then I came across another sort of diagrammatic explanation, very simple from Nature, the journal Nature. So how COVID-19 tests work. So, the two kinds of coronavirus tests look for viral material, that is nucleic acids and antigens, and a third is based on antibodies. So how does it work? The nucleic acid-based test, basically this is the PCR we are talking about. And of course, there are various others that are in the pipeline, but let's just refer to the PCR because we are all comfortable with the real-time PCR. So that detects viral RNA, and the antigen test would detect proteins on the surface of virus. And when I was skimming through the different tests, of course, the spike protein comes on top, but certain other proteins also used for this antigen test. Then the antibody test, you have to take blood. Now both these you will see are based on nasal, nose and throat swabs. But for the antibody test, one has to take a blood sample to detect the antibodies against the virus. So how is it done? Very quickly, for the RNA real-time tests, PCR test and the antigen test, the nasal or a throat swab is taken, or both actually. Where I was working, we did both together. We took a nasal swab as well as a throat swab and put it into the same RNA extraction fluid or the VTM. And then at the beginning of the PCR, the RNA that was extracted was first converted to DNA via RT, that is transcription. Okay, so RNA was transcribed to DNA first, and then the reagents were put in, and the PCR amplified or increased the number of copies of a particular area of the RNA, and that was very clearly indicative of whether the patient was positive, the individual was positive or negative, carrying a virus load or not. So this, as we all know, PCR is like photocopying. You have one copy and then you go and photocopy 200, 300 copies, and then the visibility is more. Then for the antigen test also, you take a nasal or a throat swab, and then the sample is added to another buffer, and then that drop of that is put onto the rapid antigen test cartridge, can be strips or a cartridge. The strip can be mounted on a cartridge, and this is very quick, and it can be done at point of care. Although I do not agree that it can be done by non-experts, but this is what this diagram says. Then for antibodies, you have to take a finger prick, blood sample, and again put it into the antibody test cartridge, and then you add some more buffers. And actually there are two lines here, as you can see. So one has to be there anyway, so that it will tell us that the test is working properly. And if the second line comes up, this means the person is positive for the antigen, and this person is positive for the antiviral antibody that we are looking for. Then what does the test tell you? So the PCR would tell you whether any viral RNA is there, even at very low levels, and whether the antigen test would tell you whether the virus is present in high concentrations or not. If it is positive, then obviously you are very likely to be infectious. And then the antibody test will tell you not that you are acutely sick or that you are infectious, but that you have had the virus, because it does not detect an active infection. So time and cost wise, those who do the real time, we know that we can do this, the extraction and the run within safely 4 hours, but we would say, we can't release the results. We used to do it where I used to work within 6 hours. But for safety, we would say 12 hours or generally speaking 24 hours. But we know that these days people are complaining saying test results are coming even after 4 or 5 days. Depends on where it is done and the mechanism that is used. Of course, it's very expensive, about 8,000, 9,000 rupees per test. Then the antigen and the antibody test will take only minutes to tell you the results. And of course, it's much, way cheaper. But I'm sorry, I really don't know. We can discuss this. The cost of this, I am not aware. Then when you look at the reliability of these, okay, this is a very basic presentation done by nature. So the PCR or the RNA detection tests are very sensitive and very specific. But in the case of the antigen test, this can miss infections with low virus levels. Even if you are at the beginning of the infection, you might not be picked up. But the antibody tests are also variable because the test quality, there was a question initially, but now of course they say that there are robust tests. Now let's come back to the review. And here they talk about first. So let's go through these 3 different tests again very quickly with some more depth. So the molecular testing, usually we talk about a real-time PCR. I'm not going to talk about real-time or PCR here, but this is a multiplex. We are the results that we are getting from a multiplex real-time PCR. That is to say, at one time in the same tube, 2 to 3 genes will be detected, which will make the test results more reliable. So multiplex is that. And nowadays, this is thought to be the gold standard, the real-time PCR. And this is that detects viral RNA is more sensitive for detecting exposed individuals as well as those with an active infection that shows people who are showing symptoms. And the results, of course, are highly accurate. So you can test to see whether you will be getting symptoms in a couple of days because very low viral loads are also detected. But on the other hand, if you go to the other end of the curve, right, you may be positive with the low viral load, but maybe you have gone through the entire infection, maybe asymptomatic, and you might not be infectious. So here, again, very quickly, a healthcare provider or a healthcare worker will collect mucus from your nose or throat, pharyngeal or oropharyngeal, using specialized swabs, a specialized swab rather, and the major target genes are many. We'll talk about that very soon. So before we come to the actual targets, let me very quickly, out of my interest, I looked for the schematic presentation of the SARS-CoV-2 genome, and it is a fairly big genome for a virus. And they say that these coronaviruses usually have large genomes compared to other viruses, and the SARS-CoV-2 has the size of it is 29,903 nucleotides. And this is the paper. And you can see that there is the open reading frame 1A, open reading frame 1B, and then the spike protein, right? Area coding for the gene, coding for the spike protein, envelope protein, the membrane protein, nucleocapsid protein, and there are a few others. So this is basically this single standard genomic RNA. This is the schematic presentation. So coming to the amplicon targets or the targets that we look for in our PCR. So here in this paper, which is Common et al. 2020, they have picked 3 areas: the RDRP gene, which stands for the RNA-dependent RNA polymerase, then the N gene, that is the nucleocapsid gene that codes for the protein, N protein, and then the E gene that codes for the envelope protein. So combination, this was in 2020. So now, of course, there can be others coming in, but these are the three basic ones that have been used for in real-time PCR. Then moving on to rapid antigen test. So these will detect the viral protein. So primarily it was the uh, spike protein, the glycoprotein, of course, now the M protein, the membrane protein, and then this N protein, the nucleocapsid protein, is supposed to be released to the outside. So when it is released, then through the mucus, we can detect it via the antigen tests. So this usually should be administered by trained people, but it's much cheaper, and the turnaround time is very short than the PCR test. And again, let me reiterate that this detects individuals at peak infection. So if I take you back to this simple sketch, so the rapid test would be detecting people with high viral loads, and these people are infectious. So effective for, therefore, community screening in large numbers and detecting those who are at infectious, that is, who are at greatest risk of spreading the disease. They may be asymptomatic, that is, they may not show symptoms, because we know that more than 70 to 80% people approximately are not showing symptoms in this case. So we can catch them via this rapid antigen test. But the disadvantage again is they are less sensitive than the PCR, because the PCR can pick up minuscule or tiny amounts of the SARS-CoV-2 virus. But here you can see that the range is such that they can pick up people who are not infectious also. So again, antigen test can miss infectious people, which can lead to resulting in outbreaks in countries that have largely controlled the virus transmission, which is a negative point, disadvantage of this antigen test. But on the other hand, the lower sensitivity, that is a high viral load, is required to get the antigen test positive, is considered to be positive, because some people who actually get a positive result from the PCR test who are infected, but they will not spread the virus to others anymore if you take them at this point in time or from this point in time. So, therefore, antigen tests can shift the focus to identifying the most infectious people. Then moving on to the antibody test. Now, this is a little iffy. This is again based on an ELISA. Those who know ELISA will understand this. And even the antigen tests are primarily based on an ELISA. It's also called the immunochromatographic tests, ICTs, when they are brought in or presented as strip tests or cartridge tests. So antibodies takes several days to develop after the infection starts off, but these antibodies can last in the blood for weeks after the person recovers. So therefore, serological or immunological tests, antibody tests should not be used for diagnosing the acute phase of the disease. Usually within the 7 days from the onset of the symptoms, this will not be useful at all. So antibody tests, they say, mostly have a complementary role to the PCR test in diagnosis, and usually it should be applied at approximately 10 days or more after the onset of the symptoms of a person. So actually these antibody tests can be used to detect possible previous infections in people who are recovering, or it's called the convalescent phase, considering the period of 14 days after the onset of symptoms. So antibody tests can be useful to go back in time and retrospectively assess outbreaks, or to do epidemiologic studies. You can do screening for cases that were there before and as well as to diagnose asymptomatic patients. But again, there is a problem because they have to, although they may be asymptomatic, they should be have had the infection and should be at the tail end. And then also to confirm close contacts of confirmed cases. But there again, as you can see, there is a problem. So these are the 3 tests. Then I'm coming back to the review. Now, let me introduce the term translational research. So translational research is taking research from the bench to the bedside or from the lab to clinical use. So in this situation, we are talking about development of new diagnostic tests, development of drugs, and or vaccines. So here, this is advancing translational medicine. It's called translation medicine because obviously we are taking it from the research lab to the clinic and therefore in medicine practice of medicine. So this is a schematic overview of the key innovation drivers on this side in the pink boxes, technologies, institutes, partners on this side needed for development of new diagnostics, drugs and now vaccines. Now let me take each side separately because I want to drive a few points here. Now when you consider the boxes on the left, these identify some of the most important medical, scientific and industrial deliverables. Scientific, where there is an interconnected approach that covers each step from fundamental research, say from discovering an antigen, or identify, not discover, but to identify antigen, to commercialization of an antigen, rapid antigen test. So there has to be an interconnected approach. Therefore, this interconnection is of multidisciplinary teams, where we will have life scientists that who will be able to translate basic science into changes in clinical practice, and with the support of expertise from engineering, say biomedical engineers, then we need lawyers, financial experts, and so on. So let's see who are required for this process. So to characterize the disease or to identify the genetically and biologically identify pathogen or the positive agent and the symptoms and so on. So we need scientists from life sciences, we need medical scientists, we need public health people, and so on. We need pharmaceutical scientists to identify drug targets and so on. Then we also may need engineering sciences to innovate new technologies. For example, if we want to have a platform that is like, it can be nanoscience, it can be bioengineering and so on. So that then if we come across intellectual property, if we have a new antigen test or something we want to, with intellectual property, we want to get a patent or a trademark or whatever, there has to be lawyers involved. And then funding, so finances, people who are involved in finances has to be involved. And in new ventures, we have to management of entrepreneurship. Now, a fine example in Sri Lanka is, when you take the swabs from the patient or the individual, you have to put these swabs into a virus transport medium, VTM, and that we used to get from outside, imported. But now a local company, 2 companies have come together and they are manufacturing that, and they have saved millions of rupees to Sri Lanka because of this entrepreneurship. So new ventures. And then benefit risk assessment. So we have to do pre-clinical research. We have to do clinical research after that. We have to look at the regulations. We have to look at the health economics. Health economics is how much money are we putting in versus what is the benefit coming out of it. Cost-benefit ratio. Then we also have to look at humanities. That is the social sciences. Socially, how would people, react to these things? How do they, as it is, there are psychologists coming and talking on TVs. There are social scientists saying, there is more gender-based violence now, children are depressed and so on. So they also have to be active. So when you consider the other box, these are innovation drivers, and the current innovation drivers combine the ability to process large amounts of data, big data. So bioinformatics data management, we know that we will have lots and lots and lots of sequence data, for example. So all that has to be managed. And then secondly, patient access and biobanks. Now I would like to talk a little bit about this, because if you have to do research, you have to have access to patients. Now most of us, including myself, we do not have access to patients to do whatever that we want to do. It's very difficult. We have tried, not that we haven't, but it's very difficult. So neither do we have biobanks in this country. Biobanks are where, now if a particular group or 2 or 5 groups are working and they extract RNA and they want to have a bank, maintain a biobank, either at -80 or liquid nitrogen. So that's a biobank, right? So we don't have those. That's a repository basically. So if somebody is the same patients who are, whose RNA is there, they might have the serum of these patients to look at antibodies, and there should be biobanks, but we unfortunately do not have this. So that is very important to facilitate access to biomaterial, to easy access to a biobank. And then there has to be return on investment, there has to be industrial organization. This is all management now comes into play, and collaborative innovation team science. So that is what is the other thing that I want to harp about. Now they say their very words are intense collaboration has to be there between academia, that is the universities, the researchers, and the industry. Intense collaboration. So then we must have sharing of research goals, these two parties, and directions during this pandemic. And how? Through the identification of optimal collaborative approaches that will capitalize on the strengths of both academia as well as the industry. Because once we have a test in our hand, we need the industry to mass produce it and to market it and so on. So we have to capitalize on the strengths of both, made possible for the rapid development of new diagnostic tests, drugs, and vaccines against COVID-19. Now, I'd like to harp on 2 features where once you have made a kit or a test is in your hand, you have to validate that test, or you have to look at the clinical performance of that test. When you take it to a point of care place or a hospital, how good is your test going to be? So there are 2 aspects or 2 parameters that we have to concentrate on: specificity of the test and the sensitivity of the test. So let me very briefly explain these to those who are not familiar with this. Others, please bear with me. So the analytical specificity of a test, in the case of COVID-19, let's say, is its ability to exclusively detect what we are looking for. Let's say we are looking at the virus RNA. So it has to be the ability to exclusively, very specially, pick up the virus RNA that we want to measure, for example, in PCR, even in the presence of various other targets which are not relevant to our test, right? For example, we may get human DNA. So this test has to be able to pick up specifically the viral RNA, or also to disregard various other substances that may interfere with the rest. And this has to be done under well-controlled laboratory conditions. So that is the specificity of a test. And I took the example of the COVID-19 RNA. Then the sensitivity of a test is, for example, in the same assay that we have been talking about, it describes the lowest amount of the RNA, viral RNA. Now, analyte could be anything that we are analyzing. It could be the viral RNA, it could be the viral antigen, it could be the viral antibody, but the sensitivity is what is the lowest amount that our test can pick uh, of the viral RNA, and that can be accurately measured through our assay. So these are the two. So lower the sensitivity, higher the sensitivity means lower the amount that we should be able to detect of whatever that we are looking for. So these two parameters are very, very important, and sometimes you have to work very hard to get the relevant specificity and the sensitivity of your test uh, come to that acceptable cutoff where it can be then test for clinical testing, sent for clinical testing. Then this is a lot of words, but this is just again the same thing. I'm only going to focus on the highlights, so don't worry, not the bold text rather. So defining the clinical validation of diagnostics test. So you have to assess the performance of the test, right? So that is the specificity, sensitivity. Basically, there are other parameters too in comparison with the reference test. But in this case, when you have something new, it's very difficult. But now we know at least we can look at the real-time PCRs that are very robust. So this has to be ideally done in multiple labs, not in one lab. And it has to be confirmed that the method that we are putting out is fit for use through providing well-recorded, validated protocols, the methods, and that will give good outcomes. There is a section also in this review on COVID-19 testing in low resource settings. And the key drivers they mention under these difficult circumstances would be the following. So whatever that the tests that we are proposing, or even the vaccines, they have to be affordable, sensitive, specific, the two parameters that we talked about earlier. It has to be user-friendly, it has to be rapid, it has to be strong enough, robustness, and the test has to be free of equipment and should be easily deliverable to the end users. That is, like if something can be sent out at room temperature, that is really good. Refrigeration, again, there will be problems, but even the rest can be managed perhaps. But deep freezers, deep refrigeration is going to be difficult. Then another thing is environmental stability requirements. How stable would this be under our environmental temperature, humidity, and so on? Stability testing is when you open a package or when you have filled a test tube and leave them on the bench in a humid tropical environment before we receive the samples, how stable would the test reagents be? Right? So all these have to be taken into consideration, and WHO always says that it has to be accessed by universal access has to be there for testing. So these are very important parameters. And now in the review, they say that immunoassays detecting COVID-19 antigens or antibodies are still being validated, but we know that some are validated and some strong, robust tests are out there in the market now. Then another aspect that they point out is the impact of new digital technologies under this COVID-19 pandemic. So during the pandemic, digital technologies became very important for both social health and economic performance. So let me take you through this. So there were rapid developments in the field of artificial intelligence, AI, and machine learning, for example, to screen populations and to assess COVID-19 risk factors. How many are not wearing face masks? How many have been exposed to known patients? How many are with fever, coughing, and so on? And this, they say, is really the first time in the history of pandemic or acute infectious research where AI, or artificial intelligence, and machine learning were very deeply or prominently used from the initial discovery of the virus onwards. And in China and like Singapore, South Korea, these new AI-powered smartphone apps have been used to monitor the health of individuals and track the geographical spread of the virus. And these apps aimed, it's very interesting. I found it very interesting. They are able to predict which population and communities are most susceptible to the effects of the infectious disease outbreak, to enable this, they aim to enable patients to receive real-time information from their medical providers, to provide people with updates about their medical condition without them having to visit a hospital in person, and to notify individuals of potential infection hotspots so that we could avoid such areas, and etc. So I think we also have now, I know that when not for this virus itself, but people with other various non-communicable diseases, they could send their WhatsApp their medication lists, prescriptions, and they could get those delivered to home and so on. So we also saw some of this. And then moving on, the review illustrates the public health impact of the pandemic. This was, remember, only up to September of 2020, right? So this was just the first wave. So you can see the Americas standing tall, then Southeast Asia coming later on, Europe, then the Eastern Mediterranean and the Africas, and then finally the Western Pacific. So here they say 11th March 2020, that was the day that the WHO declared this as a pandemic situation. And then, so they are talking about the first wave, and then they predict a second global wave, maybe third, fourth global waves, hope not. And they, of course, resolved by vaccine and treatment. And then, along with that, laboratory medicine consequences. So please bear with me, because this is very relevant to some of us, and the others should be aware of all these. So we know that when it's very important to sequence the genome of these viruses, and I understand constantly do that because new variants can come up. And talking about variants, when I was looking up the WHO websites and other references that were also included in this review, initially there was, of course, the Chinese strain, which is called the East Asian strain, and later on it mutated a little bit, and it went all over the world. And then subsequently they talk about a Danish variant that came up, and then of course the South African variant, and the recent UK variant, which is very bad. And recently there have been reports of the Oxford vaccine not being efficient enough towards the South African strain, and it has been pulled out. The Oxford vaccine has been pulled out of South Africa now because the efficiency is about 30%. So therefore genome sequencing and then subsequently looking at the clinical assessment of patients carrying those sequences are very important. And I saw Professor Neelika Malavige on TV the other day saying that they are constantly sequencing samples that they get to look at for variants, because variants can be of two types. Variants can come from outside, they can be imported, so to speak, or the local strain that is going around can itself mutate. So then the test, we are talking about tests and drugs and vaccines here. So we know that emergency use authorizations were given by FDA for multiple tests, and then they continued from a formal FDA authorization. And this is Central European accreditation, because remember, this is published in Nature Reviews, and that is a UK-based publication. So they are talking about Central European accreditation. So molecular tests, when you take, or the antibody or the antigen test, serological testing. So it's very important. There are lots of steps involved. So you have to develop all the reagents. For example, in the molecular testing, primers, probes, the assay has to be designed, internal controls have to be developed. Likewise, the antigen antibodies have to be selected, they have to be produced, then the test has to be designed, and in both optimization, possible automation has to be looked at, Then we have to give it to the industry. Then finally, clinical validation, right? Clinical testing. Then we can launch the test and launch the point of care tests. And then the test has to be made available. So there is what is known as progressive test implementation. So the industry has to mass produce it. If it's a vaccine, we always see how, the vaccines are made and, going in down a belt and so on. So mass production. Then there has to be extensive transport, broad implementation of whatever programs, detection programs, and diagnostics or vaccine programs, and aggressive search of the virus and destroying the virus, and then storing whatever the kits or the vaccines for future use and so on. And so all this will impact on the waves that we are expecting or that we hope to stop by having vaccines and various treatment modes. So in conclusion, they bring up 3 points. They say for future waves of the COVID-19, the current diagnostic tools should be able to be used immediately and better in a more quantitative manner, will enable the rapid detection of new infected individuals, and they can be then isolated, and confinement measures can be implemented based on the diagnostics. And then of utmost importance or critical importance, they say, is that the diagnostic tests have optimal value only when the community is fully engaged. That is, when the community is fully responsible for what is happening, and individuals will comply or listen or heed to what is being told to them, and they will participate in confinement measures, and of course they will adequately use personal protective equipment and social distancing and so on. And finally, they say there is a need for global solidarity, that is, towards test access. Everybody, every individual on this earth should have access to testing. And importantly, infection control, diagnostic interventions, that is diagnostics and infection control, has to be strongly combined or intertwined to optimally combat these current and future pandemics. So diagnostics is very important. And finally, diagnostics should be the guide to choose the mode of therapy for an individual. And of course, there has to be follow-up of therapy success. So after a patient is taken to IDH with diagnostics coming in handy, diagnostic interventions, then that person will be, of course, looked after. There will be given, therapy will be given appropriately. And then after therapy also, the success of the therapy also has to be measured using diagnostics, because I know they use the rapid antigen test, I think, to discharge. Some places use the PCR, and there are in industry and various workplaces, however much the person has been treated and has been in isolation and has passed the 14 days and how many days before they come back to work, they have to have a PCR, but that is a company policy. In the university, I understand that if the health inspector of that area of residence of that particular individual who was infected, if that person can give that person needs to give a clear certificate, a certificate clearing the individual is safe enough to come back to work. So thank you everybody for your attention. I don't know how good I was, but if there are any questions, I will be happy to take them. Let's see whether I could answer most of these. Thank you.
Thank you very much, Madam, for that interesting and very informative presentation. So now it's time to discuss this article. So if you have, as Madam said, if you have any questions or comments, you can raise them up now. Especially, we highly appreciate if undergraduate students can come up with questions and comments to convey your view on this article. So if you have any questions, please ask them now.
Actually, this is just compliment than a question, madam. Thank you very much for explaining this very beautifully. You explained this. To be honest, I am not very much familiar with this kind of research. Sorry. So you explained very beautifully, and it gave us very good awareness about this all these testing mechanisms. Therefore, thank you very much for that. If I ask a very a quick question, so you mentioned that we need to take like a nasal or throat swab for testing. But I would like to know why, when we do testing, why we need to take both nasal and throat swabs in Sri Lanka.
Because it's like this, I think the nasal swab is the better one. But the virus, as Professor Nilika Malavigi has pointed out on several occasions, the virus shedding can be different from day to day. So in order to capture whatever that we can tap, we go for both nasal and throat swabs.
Right.
That's the reason. And actually, as I told you, where I was working earlier in the private sector, we used to take two swabs, both nasal and the throat swab, and put it in the same VMT. But earlier we started off with the nasal only, but subsequently the throat came, and now I think in most places they do both.
Thank you very much, madam.
Okay, you're welcome, and thank you for your kind words, whoever you are. Any more? Have I put you to sleep?
Naomi here.
Yes, I am.
Madam, I just wanted to thank you because this field is quite unfamiliar for us, but your presentation make us much more information on actually that COVID transmission and that the things that really needed to be aware. So I just wanted to thank you for spending your valuable time for this, and it was very useful and helpful for us to to get the information like those. I believe the public should also be aware about this. Yeah. Thank you very much, madam.
Thank you. That's why I made it very, very simple. I was wondering whether these IIMB big people will, go to sleep, but then I have to cater to everybody, and I'm really happy, Iomi, that I did spend a lot of time on this, that I have been able to tell you all something in simple terms. Any questions? Anybody?
Madam, Professor Inoka has sent us a message saying his mic is not working. He said, thank you very much for the informative presentation. Question is, what are the deterministic factors to choose the diagnostic mass screening methods for a population? How necessary is it? Is Sri Lanka taking the right steps towards screening?
Yes, first of all, as you and I believe, I think we should increase the number of tests. That is number one, especially the PCRs. So I think community testing, as they are doing rightly, the community testing they are doing with the antigen test, that's fine. The PCR tests have to be widened. The numbers have to be increased. And I guess you can drive through to the private sector and, get a PCR done. So I think we are doing okay, except for the fact that, as you and I both know, after all the attempts that you went through, the hardships that you went through to put up a lovely lab, we are not getting samples. So I think we are going in the right way, because antibody tests, I'm not very sure as to how we are going to be doing that, or whether tests are being done here. I am not sure, but I know that antibody tests were brought in. But the antigen and the antibody tests are being done, I think, in the appropriate way. And of course, if we can do, I think even in universities, we should be doing random antigen tests and not just, looking at people. I have a feeling that they're sort of selecting out populations and not approaching others. So I think it should be a community-wide thing. That is one thing. PCRs, of course, the PCR testing numbers have to be increased in Lanka. That's what I personally feel. Antibodies can come in later. or after wave is over, we can, look at that, and then we can see how many were actually asymptomatic but have got the for epidemiology. I think the epidemiology unit should be interested more in the antibody testing.
Thank you, madam. I have a small question, madam. So you nicely explained about the sensitivity and specificity of a diagnostic. So talking about the specificity in this article, did they consider, like, did they discuss the cross-reactivity of these antigen, the diagnostic test with other similar
Right, they have
coronaviruses?
included some regions which are common to the coronaviruses. If we take a real time, common regions are also included, whereas specific tests are also included. Can you hear me?
Okay.
Yes. So
Yes,
they have
madam.
balanced it out, Mahishi, by looking at now even some of the antigen tests. I think they have three bands. One is the common corona band, and the other one is the specific COVID-19 band.
Thank you very much, madam.
Madam Sujanti here.
Yes, Sujanti.
Thank you for the interesting and informative presentation. And I have few questions. And I heard that in some countries, so India or somewhere, they are using COVID-19 test kits at home.
Yes.
What sort of those kits, what type of diagnostics are those kits?
I have a feeling those are antigen tests. Those must be antigen tests. I'm not 100% sure. I did see it, but if anybody else knows better, why don't you speak up? But right now, for now, I know that there is, even in the West they have, and even in India, but I feel this is like home pregnancy test, something that is simple. So I feel that it is the antigen test.
Okay, madam. And then, madam, you told that in the structure of the morphological structure of the COVID-19, there are spike protein, and that is important for the invasion into the host. And how come it, madam? Because it's going through the nasal and throat to the lungs, so it is for. Are you referring to the that is invading the lungs? So
Invading the mucosae of the nasal and pharyngeal passage, right? So because they can't just pass through the nose and the pharynx and into the lungs without living first and multiplying. So they will enter the nose, right? And the pharynx, the pharyngeal oropharynx, and then they have to colonize there first, right? And then only when the numbers increase, they will go down to the this is the mucosa, the epithelial cells, so to speak.
okay,
Mucosal epithelium that they're invading. And then finally they're going to the lungs.
Okay. Thank you so much, madam.
Okay.
Madam Thanusha here.
Yes.
Madam, I have a small clarification actually. Now, as you said, like the high mutation rate of these coronavirus, how would be this sensitivity of these our test that would, like, because now this is like really this has a high mutation rate rather than the previous invaded viruses that we faced throughout. So this has, like, within a very short time, this is like we came across a few strains.
Yes.
So how would
so
be?
like
Yes.
Yes, so they will have to come up. Now, usually these tests are based on, like the PCR is not a problem because you have the conserved regions as well. And then even if there is variation, I think the spike protein is the one that varies mostly.
Yes.
And I had a question in my mind also, if a rapid antigen test is based on the spike protein, and if the spike has, but then in the spike also, the entire thing, they are not going to, there will be some epitopes, right? Some epitopes which will be there, only certain epitopes, if you know what I'm talking about, certain regions of the antigen would mutate, not the entire antigen. So, but I think that is why it is extremely important for scientists to be constantly looking at the strains that are going out, and that information should be deposited in the NCBI or some place where they can access that, and so that the industry, scientists who are associated in the industry can act appropriately. You are right. That is why I told you this, the South African strain is not working for the Oxford vaccine. Oxford vaccine is not good enough for that.
Yeah, and madam, that should be it can be a dependent factor as well, because now it's in most of the Western countries, they have access for all the to do the quick screening and to sequence the viruses, but in other countries, by the time they're getting familiar with this new strains, or might be that could be another factor, because as an example, if we come up with a new strain, I don't know how, with the facilities and the extent that we can go
I think, I know there is a big question there, but I think we are okay. I'm not saying we are 100% okay, because even with all the technology that the US has, see what has happened today. So on the other hand, when the UK strain came to Sri Lanka, we detected it. So we are not that bad, I feel. Of course, India is much better, although we laugh at them all the time. I think there is a balance. Of course, we might miss cases. I'm not saying it's a 100% blanket situation, but I think we are doing what we can. I think our scientists are doing what we can.
Yes. Yeah. Thank you very much, madam. Thank you for your nice informative presentation as well. Thank you.
Thank you. Who?
Madam, can I ask another question?
Yes, sure. Who is this? Can you identify yourself, because I can't see you guys?
Madam, I am Ashoka Ranjiva.
Yes, yes.
Yeah. Madam, do you think that Sri Lanka is doing enough rapid antigen tests?
No, I don't think so, because I think if you take the university crowd, if I had authority, I would, do better than that, because we can, I think we should do better than that.
Yeah, because we don't know much what is the community spread, and we can, if we do lots of rapid antigen tests, we might be able to identify the community spread throughout the country.
Yes, absolutely. Now I'm focusing on the university, for example. Has anybody come? But actually, the antigen test is now available for the campus. But I don't know why they are not using it even randomly, because with these students coming in for exams and so on. But from the communique that we got from the vice chancellor, I think they have come up. The management has come up with a very sort of a good plan, that antigen tests are available, and if anybody comes positive, they will be isolated. There has been a hostel dedicated. I'm just talking about the university. Okay, they will provide facilities for isolation and quarantine and all that, and with doctors in attendance from the faculty, and all that has been informed to us. I agree with you, Ashoka. Obviously, of course, antigen tests must be costing. And I think, but I think we should be more proactive in that sphere.
Yeah, I think, because we are much focused on PCR, which is more expensive than the antigen test.
It might not also be telling us exactly, whether they are infectious or not. On the other hand, so I think at the primary level, as you say, antigen testing should be, we should see a more broader use of antigen tests in the community.
Yeah. Thank you, madam.
Thank you, Asuka.
Madam, this is Vidya.
Yes, Vidya.
Thank you so much for the very lovely presentation, madam. As like everybody said, it was kind of, you generalized it really well so that everybody could understand it. Madam, I just wanted to know, this vaccine that many of like some of these people in Sri Lanka got, like it was supposed to be of 2 doses.
Yes.
Maybe this is out of this review paper, madam. So now there is talk that only 1 dose is enough, something like
Yes,
that.
they say that. They do say, earlier they said 3 weeks apart. Then they said it should be even 4 weeks apart. And I don't think it's this vaccine. I think it's the Russian vaccine that they are saying 1 dose.
Sputnik.
Yes, because that is supposed to be 93% efficient. I actually didn't concentrate, actually speaking. Can you remember Sakuni? Sakuni Ram.
Yes, madam.
She had done a presentation to her lab group at Houston, and Preethi Udagama, my friend, sent me that presentation, right? And that actually, even if we can connect her online, that would be really good even for the IIMDs. Gives a lot of information about all the different vaccines, comparing the vaccines, the efficiencies, how they were made, and so on. I do have it, but I thought it's too much to sort of even get permission to use her slides. I thought I was, because this is just focused on this paper, but yes, we don't know enough about. And actually speaking, my son asked me to get registered onto this vaccination website. I'm like, okay, which one am I getting, and am I happy with it, and that sort of thing. So we should know more about it, more about vaccines actually. Yeah.
And the immunity that it gives, madam, like
People actually don't
for
know.
how much time?
I mean, they say 1 year, India, but we have to wait, watch, and see. Because I don't think within such a short period of time you can, talk about the entire immune responses and for how long they will last, and whether this is protective or whether actually, usually what we do is we simulate the natural protective immunity through a vaccine, right?
Yes.
Natural immunity, there has hardly been any time to sort of follow up.
Evaluate
Yeah,
what
yeah,
happens.
yes. So I hope I answered your question, but that's the best I could do. I'm sorry.
Yes, madam. Thank you so much.
Madam, Sachini here.
Yes, Sachini.
Madam, thank you very much for the very interesting speech, and that matches with the broad audience. And madam, I just have a small question. I don't know whether I have missed this. Are we doing antibody? Have we started doing antibody test in Sri Lanka?
I heard that the antibody test has been imported, has been brought in, but I have not heard or seen anybody using it. So Sachini, I don't know how to answer your question. I think the epidemiology unit, as I feel as an immunologist, the epidemiology unit should be responsible for that part. We can start doing that. Because
madam, like what I feel is this is just my idea. So like especially when you take these symptomatic patients and non-symptomatic patients, if we miss bleeding them at first, like within 1 week or like 1 month, then we'll be missing some information
that's the
at
thing
the
now.
starting point.
That now the Navy cohort would have been lovely, some of them were sick, some of them were asymptomatic, right? So as a researcher, you know the problems that we face. No, we don't have access to them. I mean, like, so that cohort would have been really nice to have done an antibody survey. You're right. Yes, you're right. I don't know whether anybody is doing it, but to my knowledge, which may be limited, I have not heard of anybody doing antibody tests such. And we did try to tap into that Navy cohort, but it was not possible. We tried very, very hard, but we couldn't.
Madam?
Yes.
I have a question. It is not entirely based on this paper, but kind of related to PCRs. Like I have seen some articles that mention there is like viral COVID-19 RNA in fecal matter and some water bodies. So as I understand it, it does not.
Yes, sorry.
Yes, ma'am. As I understand, it does not imply the infectivity, right?
Yes.
Of the Is there any way to assess that, madam?
I think by now, okay, by now we should have, the sketches that I showed you. I tried my level best to get at the article that would tell us, okay, this viral load and you're infectious, this viral load and you're infectious is not there and so on. But I'm not saying I tried very hard. I even tried to contact Malik series, I couldn't. But I think the viral load should tell us something, don't you think? Because in PCR, usually we just say positive or negative. That in certain labs, they give the viral load as well, right? So higher the viral load, I mean, by now, we should be able to have plots of people who are infective versus those who are not. Those who are asymptomatic, those who are not. These are lovely studies, but maybe these are published. I'm not sure. So likewise in wastewater. Actually, we have been proposed a project where to get involved in doing PCR for wastewater for COVID-19. And I think urine also sometimes can be used, but blood, we are using blood for PCR. That means it has come to the systemic blood, the peripheral blood circulation, which means that the patient is, really bad, doing very bad with a very, very high viral.
Yes, madam. I have seen another article. Thank you, madam, for the explanation. I have seen another article that like argues there can be like if virus is active in fecal matter or in like soil, the animals can like it can re-jump to the animals. So
that is news to me. Can you elaborate on that, please? Yes. No, I don't.
Yeah, it's just an argument, madam. I don't know the like validity of that. I was just curious.
I doubt that very much. The way people are, defecating, and then India must be, I don't know. I mean, I really don't know. But that's an interesting point. Thank you. We should look into that.
Yes, madam. What I thought is if we can like maybe culture the PCR positive whatever material to see they like contain actual virus that can replicate or infect.
You're right, because even pieces of the RNA can give positivity, right? Which might not indicate a live virus. But well, yeah, yes, your suggestion, I think, yes, it's well noted. Yes. But I doubt whether from soil we get it. I really don't think so. From saliva, they say one can get it. Nasal secretions and droplets, but they do not talk about, but they do, you are right. Yes, yes, yes. Because that is why washrooms were allocated to separate groups. Yes, it can go through. Yes, you are right. Yes. But no elaborations have not been done on that, right? Those are reports. Those are those scientific reports.
Actually, I'm not sure, madam. I just saw as an argument.
Like, okay, all right. I have not seen any concrete scientific evidence coming out. From the literature that I read right now, what they say is saliva, nasal secretions, droplets, airborne droplets. Those are the ones, and of course, contaminated objects for a certain amount of time. Those are the sources of contamination.
Okay, madam. Thank you. Thank you for your.
Thank you, Putra.
Madam.
Yes.
Madam, I'm Indivari here. Madam, thank you for the interesting presentation, madam. So, madam, as you emphasized PCR to be the gold standard in diagnosing, and also the molecular biological approaches along with the immunological approaches are like applied in parallel in order to avoid the false negatives. So in that light, madam, I would like to know the applicability of the clinical diagnostic methods such as the computer tomography and like ultrasonic imaging, along with the routine diagnostics for the validation of the results, madam?
Well, as far as I know, oh, that's tough. Okay, so you take a patient and you have symptoms or you believe you have been infected and you do a PCR, and if really if you come positive and if you are in that infectious range, I suppose you are hospitalized, and if you have non-communicable diseases on top of that. Otherwise, I think just being simply positive is not good enough in certain countries for you to be admitted to hospital, because you are asked, especially I know in UK, they ask you to stay at home with a certain viral load. They ask you to stay at home and they constantly check on you. And even in Sri Lanka, I don't know whether I know of a case where she was positive, but she never went anywhere. She was at home and being a doctor also. So I have a feeling these complicated cases are only hospital. I don't know. I think otherwise you are sent to isolation centers or quarantine centers. But the bad ones are the ones who are hospitalized. And in the hospitals, yes, they are constantly checking you for leakage and this and that and the other. But I think the doctors are able to categorize the people, right? The community physicians or whoever who gets the results are able to categorize you into these different categories. And I think, Sachini, you can elaborate more on this, depending on the, is it the viral load that you are sent to isolation centers or quarantine centers and you are asked to check into hotels and so on. And I think the real bad ones are only taken into hospitals. Sachini, can you fill in?
Madam,
Yes.
that's what happens. Basically, they ask, but still in Sri Lanka, I don't know whether it is legally, I mean, accepted to keep us at home.
Yes,
because when
but I know of a case that
yes, madam.
And she was a doctor. She is a doctor anyway. So
Okay, madam. So because what happened was now in my place, both my in-laws were non-symptomatic, and we tried to keep them at home because there's no one at home, so they can easily stay there. So we requested several times, but then they did not allow, saying that they don't have any right to keep them. And then even though they were non-symptomatic, we had to take them to the quarantine center, to a private quarantine isolation center actually. And like if they have complications, when they check, when we enter there, they will send them to the hospital. If not, they just keep them there and regularly check them.
Right, because I know of a person who was admitted to IDH because of very high diabetes.
Oh yes,
high
yes,
sugar.
like yes, even my father-in-law was having diabetics, but they said to come every morning and get that checked. So if something goes wrong, they will so that they can send him to the hospital. But luckily nothing happened. And according to what I heard, they don't allow older people to go to these private places. But in our case, they allowed, yes. So I don't know whether it's really true or not.
Yeah, so it's not 100% regularized.
Yes, yes.
But for the person who asked the question, that is the rough answer that I can give you.
Thank you, madam. Madam, I have another question. Now, since like unusual specimens such as the conjunctival fluids also were found contagious, so it's like it's very important to adhere to safety precautions when handling the clinical specimens, right?
Absolutely.
So, madam, I would like to know about whether that WHO has developed some guidelines. Are there any special guidelines?
They do have guidelines. Of course, in the labs, we have to wear this N95 mask. That was from the very beginning. And then goggles and what do you call the hair caps, to cover your hair. And the aprons and the lab coats, not the regular lab coats. So lab coats are worn underneath, and on top of that, you have to wear disposables, which cover your entire, body. Only your hands are out, and the hands are also, you have to put double gloves. Okay. You have to put double gloves, and the sleeves of the attire, the coats, these disposable ones, are, what do you call it, with the elastic, right? So it has to come right up to the wrist, and so that no skin is exposed. So that is how we did things where I worked previously, adhering to WHO guidelines. I don't know whether everybody is doing that, but guidelines are there.
Madam, can I add something on that, madam? I'm Sahan.
Yes, Sahan. Yes. Go on.
Yes.
You need first-hand information. Yes.
Madam, for that information, WHO is actually updating the documents periodically. So they have amended some of the documents that they were released initially. I mean, in December and January, and they had some amendment in March as well. So they are continuously doing that. Once they have data on how the things working, so they amend the documents as well. But for that, madam, the lab coats and safety stuff, madam, it's actually they finally I mean, even in the document, they finally say that do a assessment yourself in the lab and adhere and change the regulations within a frame to match your requirements.
Okay.
So in case if we have a really good facilities and everything, so you can actually go with the usual lab coats, but you have to have double gloves and sleeves and everything, and also the 95 mask and the goggles. So it's depending on the lab you have and lab we have. We can actually, the lab technician or the highest authority can decide what we can do within that frame.
Work. Okay, right. Thank you. Anything else that you would like to add on to this? Because you have been doing it not only here, but also in Germany. Anything that you would like to tell us? Anything more?
Actually, madam, I was going to say that you have really simplified everything, and you have done like the whole thing, like we've been inside this thing, the whole thing, and you have simplified all the presentation into a certain kind that everybody can understand, madam. That is really nice, madam.
Thank you.
Just I wanted to
I would like to add anything that you would like to add, anything new, anything that I can learn from you, anything? Anything about the test kits, or now they are.
I was actually I had some questions about the I was going to ask madam whether I'm also not familiar about that. I was going to ask why we are not using the lamp method, madam.
Yeah, but the lamp method, I know it is also included in this paper.
Yes, ma'am.
If you read it, it is there. But I also am not sure, because now even for malaria, they have got really good lamp tests out there. But people are not using. Maybe people are not familiar with it, or because it's so simple, people are not believing in it. I don't know. That's a
problem, because there's a kit from UK. There's a kit for lamp method in UK, and I know a group of people that they are using. They are trying to build a unit. They are electronic people. They are trying to build a unit to use this lamp method in Sri Lanka.
Now for that, can I ask you whether the reader should be there, whether the actual small equipment should be there, or can you just take it? Can you check it out with the naked eye?
It depends on what you are using, madam. So in the lamp method, you can actually some kits that you can detect with eyes and with your own eyes, and some kits you need a reader. I think with these reagents, you need a reader, madam, but it's not sophisticated as the RT-PCR.
Yeah, I know. It's not that expensive.
Yes, madam. That's the thing. That's the question. Why we aren't progressing on that the land.
That is something. It's a global thing, isn't it? Now we also set up this Chamini.
Yes,
Chamini Kanitivala. She set up with her foreign supervisor. She
yes.
set up a lamp, a very good lamp, say for this phytoplasma, right? Sugarcane phytoplasma, but it never took off. I mean, they could have used it in the field, not the field, but
Yes, madam. I'm not sure whether there's a problem with the specificity as well, madam. That I might have to read on it, madam. But as far as I know, madam, it's the same thing. It's
the same as the PCR, but I think it's a good test because I went to workshops and for malaria, I'm talking malaria, and they were using it in Cambodia. I can't remember where I was. They were using it and that group was really, saying this is so good and all that. They had got patents and this and that, but it in our country it never took off. The anti-malaria campaign never touched it. It would be more cost beneficial to do, these random tests or whatever, but they never took it up. I don't know. That's a good question. Thank you. So in Germany, what is the situation? Did they check on anything like that? Or because actually there are other tests, there is CRISPR coming up and various other methods, but PCR is the one that has stuck, isn't it?
Yes, madam. In Germany as well, they are actually continuously, as far as I know, continuously checking on the reagents and all the things, because they are using in the RKI, they are using their own reagents. Like they have designed everything and they are using that, but they are continuously checking on the variations. And I mean, they are really serious about the on the mutations that are happening. But other than that, madam, they are really they are doing the PCRs and they are not actually like they are trying the and I mean I also tested with an antibody test. I got tested with antibody test and everything, but still they are using the PCR as the
right.
because it's yes, madam.
Antigen or antibody?
Antibody, madam.
To see if they
they are getting commercial kits to test out because I mean companies send their kits to test. I mean the validations and everything because they have the samples and they do the test and they do the validations in the institute.
Right.
They are trying lots test kits in there, madam. Yes, madam. So in there, madam, that's why I told you that the laboratories can decide what they can wear because they just wear the normal lab coats, that is wraparound lab coats, and but they use the sleeves and double gloves and everything.
Okay. I don't know at the time. I just started off in March and I was there till June.
Yes.
That was at the very beginning, 2020. So we were extremely careful because we were doing about 200 samples per day.
Yes, madam. Yes.
So we were extremely careful and everybody was so nervous, right? That we equipped them with the best things that we could. And so they had all these things. It was very difficult to work with those on though, It's very
yes, madam. But I think that's better in Sri Lanka, madam, because we are not leaving anything, to be in contact and we destroy everything. So that's, I think, better than anything.
Yes,
so
yes. We incinerated everything at the end of the day. Yes. Right. I think we should wrap up now. Dr. Maheshi.
Yes, madam. So thank you very much, madam, again for that very clear and informative presentation. And as you know, as you can see, how these participants are asking questions and comments. So it clearly shows how well they understood this presentation.
So it was my big pleasure to be with you all. Thank you so much.